सकते हैं, ठीक है?
Very well, thank you for such information. This my question is that as you know that climate change and the global warming are all results of us humans only. Ma'am, I would like to know that the interference in the nature is right that this genomics we are making this as your previous slide also stated that on users and growers choice you only make these hybrids. Previously one breeding picture was there which proved that it was not edible, it was not suitable for consumption. So, Miss, it can happen that under such pressure or something that on growers, which they make, they can make wrong genomics and wrong hybrids that can prove harmful to for consumption of the people.
I totally agree with you, and this is an extremely valid question, and there are two aspects to this. So the first is that indeed one has to be extremely careful about the research that goes into developing any kind of biotechnological version of a crop, especially an edible crop. The second thing is that there could be misuse. Whenever you develop a technology, there are chances of its misuse, and that is the main reason why we have these super authorities such as the Indian Council of Agricultural Research in India. So we have large number of policing levels for development of especially in case of biotechnological or genetically engineered crops, and it's not easy for them to just come into the field simply because somebody wants it or somebody developed it. So the guy who wants it could be a businessman. The guy who develops it could be a very, very brilliant scientist sitting in a laboratory who's interested in being able to provide something to mankind. But whether the guy who develops something is going to make it and immediately provide it to the farmers, there's no direct connection like that. So all governments, all countries, all nations have their own set of rules, and a large number of these rules are so tight and so tough that it is not possible to trickle through all the time. So I think there are enough levels of these protections that are inlaid, and yet the fact that a misuse can happen for technology should never be a reason for us to stop carrying out technology or to stop trying to make something or build something or invent something or to discover something. For example, when the BT cotton came in, a large number of people thought that in future a single gene transfer could build any kind of transgenic crop, and that would be great. But at that time they didn't realize that a large number of traits are not single genic traits, they are multigenic traits, and multigenic traits cannot be transferred to just anybody else or any other genome that easily. It's a very tough research problem. It's a very tough experiment. To even succeed once, leave alone succeeding repetitive times to build a crop that will reproduce with those set of external genes which are going to be able to give you something. That said, the second thing is that you all are probably aware of the controversies, debates, everything going on. Mutational institute, a biophysical institute where mutations or actual UV radiation was put on the plant and it was sent back to the fields, and if a new mutation was found which was good, if it was bad it would die itself, if it was good it was brought into the field. And you've probably been eating, when people across the world have been eating such plants because there were no policing going on at that time. Nobody is checking how you are making these new transgenics. Now all that is happening is now we are using very specific, very intelligent, very carefully designed experiments and very safe experiments. And I think that is why so much controversy has developed for this beautiful discovery that has happened, that we can indeed improve plants in a safe way, rather than leaving them open to UV radiations and all sorts of other kinds of mutations, which was going on for hundreds of years. So be a little more open. Be open, read about it. Don't always believe people who are not scientists that something new is bad just because it might improve mankind or lead to business for some people. I would always, as students, as youngsters, give the benefit of doubt on both sides and then make your decisions. Okay? Okay.
Morning, ma'am. My question. Ma'am, as we know that rice cultivation requires very much quantity of water. So, is there any technology developed to reduce the quantity of water used in rice cultivation?
That is another very good question. So there are different kinds of things or research projects that are underway, including looking at looking at plants which do not require a lot of water, and trying to find the genes which are responsible for enabling or giving that facility or the ability to the plant so that it does not require so much water, and then comparing them with the genes of rice. And the rice genome is already out, so a lot of research is going on in that area. And the other thing is that I think in the next even a decade, it would be very difficult, because requiring water is not the only issue. Requiring water at a specific time is an important thing for rice. So it requires water during that period, and during that period it needs to be literally waterlogged, so to speak. So there are genes which overcome water requirement, but then which are the genes, at what time will they be expressing inside the cell, and what extent do they have to be expressing inside the cell? I told you there are complications of from one gene to multiple genes. Now this level, this question that you're asking, has a complication of at another level. Once you're able to insert genes which are going to allow a certain plant to overcome the requirement for water, you would need to know at what time do those genes express. They cannot be expressing at all times. So they have to be expressing at a specific time within the life cycle of that plant. Second, they can't be, during that life cycle, at that point of time also, they cannot be expressing completely. They have to have a certain level of expression, which we have to be able to control from outside. So these increase the number of, the amount of error that can happen in the experiment. So it makes the question or that research project extremely tough, but people are working on it. Yeah.
Ma'am, as you said that insects and mites are currently destroying 13% of potential crops, and why only spider mite are able to advance new pesticides?
Okay, there are two sides to this. Again, as I understand your question, you're asking why is the spider mite genome able to overcome or combat pesticides, whereas most other insects cannot overcome or combat pesticides? Very good question. Now the thing is that number one, we are not aware whether a lot of other mites or insects can do that. To our idea, to our knowledge, because the genomes are very few. For pest genomes, we don't have a lot of pest genomes. And in fact, the spider mite was the first of its kind in its entire order. The hierarchy of animals, you have these families, you have taxa. So the senior-most taxa of chelicerates, there's simply no genome at all available for us to compare to be able to say when did this ability evolve? And did it evolve before the division or diversion of the chelicerates from somebody else, from the other kinds of animals? So I'm not a zoologist, but as I told you in the beginning, one has to study evolution, right? So when you study the evolution of the spider mite by having multiple sequences from various different kinds of animals, only then will you be able to make out that these genes which enable hijacking, cutting and pasting into your own genome, the interesting genes from other people, were these present at the time, at the millions of years ago when the chelicerates diverged from other groups of animals, or were they not present at that time and evolved much later because through natural selection, Darwinian selection probably, mutation happened and a new ability came into this organism which is more recent, such as let's say just a couple of million years ago, and therefore it has not happened in other organisms. The answer to this question can only be answered if we have sufficient number of sequences to carry out an evolutionary phylogenetic analysis of that specific set of genes, which will tell us when did this ability enter that form of life. Was it available pehle se, or was it recently procured, and when exactly? So biology has, phylogenetics for example, has the mechanism or has the potential to find out exactly how many million years ago did that specific ability happen in that organism. So it's a very interesting question to answer, and I think with the spider mite genome being available, a lot of people will be wanting to find out about this. We don't want a lot of pests to be having that ability, so we would like to be aware of who else has that ability, isn't it? That's a very good question, thanks.
Ma'am, what is robotics microarray spotters?
Okay. Are you aware of microarrays?
No.
Okay. Alright. Alright. So, where should I begin? Microarrays are a technology that allows you to see, in a very simple layman's way, which gene is expressing itself in an organism and which gene is currently lying dormant and sleeping. So some genes, not all genes in your body or in your genome are active at all times. We are humans, so I should not be comparing at all plants and animals. So in plants specifically, there are times when the plant suffers stress. At that time, the plant is able to switch on certain genes which are going to help it to overcome stress. At other times when that stress is not there, for example, have you ever noticed that a caterpillar, when it starts crawling and starting to eat the leaf of a plant, it will never crawl beyond half the leaf. At the point it reaches the half leaf, it has to jump over to another leaf. Why? Because by the time it has eaten till that point, the plant has switched on the genes which makes that leaf repel, which make the caterpillar repel that leaf, and it jumps onto somebody else where it is not aware yet. So those genes have become switched on, and they will get switched off as soon as the caterpillar jumps away, and they will get switched on in that other portion of the plant's body where the caterpillar now is, if at all the caterpillar is still around. So my point being that in plants, certain genes are switched on, certain are switched off. The ones that are switched on will later be switched off. Okay, so the time period for which they are switched on is extremely important, and for understanding any kind of stress. For example, your friend here was asking water stress. So if there's a certain plant which can grow without water and handle drought, for example, cacti are very good for handling water stress. So cacti have certain genes in them which allows them to overcome stress, and those genes are switched on when drought happens, when the deepest, worst drought stages happen during their life cycle. So how do you find out which are those genes which are on now? So if you have a list of all the genes in the organism, in this plant, in the cactus, let's say there are 20,000 genes in the cactus, you list them all out, A, B, C, D, up to 1, 2, 3, up to 20,000. Gene number 5 to 6 are on, then gene number 70,000 to 75,000 are also on, the others are off during that specific stress which you are interested in. Now the way I presented it, एक notebook पे you have these, एक gene को represent करने के लिए एक line नहीं बनाएगा, एक आप spot बना देंगे. So every gene is represented as a spot. So if there are 20,000 genes in cactus, those 20,000 will be represented as 20,000 spots. And when whichever one of them are switched on will light up like a bulb lights up. So 20,000 may say 5,000 will be green and 15,000 will be red. Red means off, green means on. Okay? So when you see this page, which in technology is a plate, it's a chip. On that chip, you have these 20,000 spots, each spot for one gene in the plant. And they switch on and off, and they change colors from red to green, showing whether or not those genes are on or off. So this technology is the microarray technology, where on a small chip, you have arrays of spots. And these are microarrays because they are very tiny. So a chip would be this size, and it would have tiny 20,000 spots for each gene in that plant or animal or whoever we are studying, mouse, yeast, E. coli. So you have chips for each organism, and each chip contains all the genes. And there are this microarray technology allows you to have the chip under stress, the chip under no stress, the chip under stress X, the chip under stress Y, the chip under stress Z. So when you compare A, B, C, D, X and no stress, you know that when the plant had no stress, these 50 genes were off. The moment stress A happened, 5 of those 50 light up. The moment stress B happens, 10 of those 50, which are different from 5 of those previous 50, light up. When stress X happens, somebody else lights up. That tells you that those 5 genes are critically important for that specific stress which you were looking at in this plant. And there are lots of other things you can do with this technology, but this is basically the microarray technology. And robotics is extreme because these are automated experiments that are running. You can't see a gene, you can't see things in the genome at all. So it's all on that chip. Somebody has to be able to see it, somebody has to be able to see the green and red color also and click pictures. and then show you that picture in a way where you can, on the computer, enlarge it and see how many are green and how many are red and how many are not affected at all. Because it's not possible that somebody will remain off all the time and on all the time. Some guys are going to be switching on and off constantly, so there'll be a yellow color also, and mixed colors. So, that is microarray data, and robotics is becoming more and more useful lately to automatically study hundreds of chips together, because then you can see hundreds of situations and compare them together. So we are not only using microarrays to understand stress A versus stress B, we are also using microarrays to understand the genes that are off in the leaf and on in the flower, and off in the root but on in the leg hemoglobin, for example. So different parts, organs of the plant are also being used. So there are thousands of situations where you want to compare the microarray chip. Which guys are on, which guys are off, when are they off or on, how many of them are off or on. So robotics is extremely important. So robotics microarray spotters are the guys who spot these microarray spots through robotic mechanisms so that human error does not come into the picture.
Ma'am, how can we use it to reduce the pests?
Reduce the pests. Okay. Lots of ways to start with in a very simple way. Now that, alright, so, okay, again, so you have the plant which is overcoming a certain kind of pest, and you have a stress resistant plant, and you also have a stress sensitive plant. Sensitive means that guy or that plant cannot overcome or combat the same stress. The stress each time is, let's say, UV radiation. UV radiation can be handled by plant A, but plant B, who is a distant cousin of plant A, cannot handle UV radiation. So the first question that you would like to ask is, what is there in plant A that allows it to overcome UV radiation? So you will compare microarrays between the two plants under UV radiation. So here you have plant A under UV radiation and without UV radiation. Similarly, you have plant B under radiation, without radiation. So I'm setting up an experiment. I'm basically designing an experiment for you. You have 4 situations. In these 4 situations, in plant A, which can overcome and combat stress, in plant A, the guys who will switch green, which were not green in the no radiation situation, but green in the radiation situation, are the genes which are responsible, probably, putatively, potentially, for overcoming UV radiation stress. Those set of genes, now you list their positions, because these microarray positions are extremely important. The first column, third row, first gene, first spot. Okay, note it down, note down those numbers here. Now you go back to the plant B and compare there also. There, between the no radiation and the radiation, you would actually not have a lot of difference, because this plant is sensitive. This plant does not have those genes which can make the difference there. But it might have some remnant, because it's a distant cousin, it might have some remnants or pseudogenes which are similar to the guys in the plant A. So there would be some people who are switched on into green, but they are unable to overcome or combat the stress. So you list down those spots also. Then you get a set of those genes and you compare those genes and you try to see, okay, this plant does not have this set of genes, and there is a series of other experiments that you design to finally be able to put these genes into that other plant. That is one way that maybe that plant will start eventually overcoming that pest. The other thing is to try to kill the pest by developing a toxin, by finding out a bacterium which has a toxin which can kill that pest, then use that toxin to put into whichever plant you're looking at. So these are very simple ways I've told you to design different kinds of experiments, but there's lots of projects that are running which can be used for using the microarray technology to overcome various kinds of stresses such as pests. Okay, does that answer anything? Have you understood at all?
Yes, ma'am.
Even if you've understood a little bit, you can probably read up later and find out more details. But if you've not understood, then it's bad.
Thank you, I've understood.
Thanks. Yeah?
It was very delighting to hear you, ma'am. As we talk from the basics, water resources are getting declining, and climate instability also leads to the reduction of productivity. So, in what aspect or in what extent gene modification is going to be useful in future?
Gene modification, see, what you call gene modification is not something that can immediately have an impact on climate change across the world. So I'm not completely sure of the kind of question you're asking. Are you saying that by modifying genes, scientists believe in changing the climate?
Actually, ma'am,
इसको overcome करने के लिए genes को modify कर दें. See, biotechnology is basically meant to be able to answer such questions only, but I'm trying to think of a situation where you could indeed do this. Now, let us say this extreme ozone holes are creating a problem for the Arctics, and there is a fear or a threat that the climate might change after another certain number of years. There is no great such threat yet, as recent research has shown, but suppose something happens through ozone holes that damages the Arctic ice and water, and the ice melts down and can hamper us. So what we would want to do is somehow cover the ozone hole, or somehow make a difference in such a way, use genomics, or my idea is not to use gene modification, but to use biotechnology and genomics in such a way as to overcome that problem. So nanotechnology can probably be used. Nanotechnology, in terms of the interaction of light with metals, can probably be used to have an effect so that the rays coming down directly from the ozone hole get reflected back. So use specific kinds of nanoparticles and spray them around and try to overcome that. So that is one way how genomic cleaning, how biotechnology can be used to overcome climate issues. If you specifically want to know how gene modification can alter climate problems created by climatic situations, then of course you would like to have more plants which can overcome waterlogging situations. Only rice or a few others can handle water, not everybody else can. So we would like to carry out such a research and preempt the possibility of plants not being able to die under extreme waterlogged situations. That's one way of looking at it. But there are like millions of things that can be thought of and done in this area. Yes.
अभी जो है ना पूरे दिन मैडम रहेगी, अपन बात करेंगे बाद में। और जो अभी आप बात कर रहे थे, आप पूछ रहे थे कि जो इन्वायरमेंट है, इन्वायरमेंट का जो प्रभाव है, इससे जो वाटर सोर्सेज डिप्लीट हो रहे हैं और जो लैंड में प्रॉब्लम हो रही है, उसमें किस तरीके से जीन मॉडिफिकेशन हेल्प कर सकता है कि नहीं कर सकता। निश्चित तौर पर बायोटेक्नोलॉजी इस दिशा में काम कर रही है। जीएम क्रॉप्स की बात करते ही हैं ये लोग। अब जीएम क्रॉप्स के बारे में भी कई तरह की बातें हैं कि वो कितने यूजफुल हैं, क्या वो जिस लिए बनाए गए हैं। पेस्ट कंट्रोल की भी बात कर रहे थे। पेस्ट कंट्रोल में भी जीएम क्रॉप्स कहते हैं कि जो स्पीसीज है, जो जीएम बनाई गई है, वो निश्चित तौर पर पेस्ट कंट्रोल में भी मदद कर सकती हैं, ऐसा उनका दावा है। पर कई तरह की समस्याएं भी हैं। तो पर लेकिन ये निश्चित तौर पर कि बायोटेक्नोलॉजी लोगों की भूख मिटाने में सक्षम हो सकती है, और जो इन्वायरमेंट में जो चेंज हो रहा है उससे एग्रीकल्चर को बचाने में भी सफल हो सकती है, ऐसा लगता है और हमें आशा रखनी चाहिए। मैं बहुत बहुत धन्यवाद देता हूँ डॉक्टर गीतांजलि यादव को कि आपने जो यहाँ पर एक बहुत ही अच्छा लेक्चर दिया, और बहुत ही सबसे बड़ी बात मैंने पहले ही कही कि जिज्ञासा के मंच पे यंगेस्ट साइंटिस्ट इस मंच